So good afternoon and welcome. I'm just going to put my video on. So yeah, that's me. Thank you very much for coming to this session of Radiopharmaceuticals. I'm going to hand over to my co-host, who's actually the main host. So Dr. Amanda Manikam, over to you.
All right. So good afternoon, everyone, and thank you for this opportunity, Prof. Welcome to today's webinar, where we will be talking about advancing access to radiopharmaceuticals in Africa, and also a little bit about what we should be doing as well. So this is done under the Pharma Connect Africa, with Prof Katerere as the TUT Chair for Sustainable Entrepreneurship, and I'm the Deputy Chair. So we have a YouTube channel, Connect Conversations Africa, where we upload the recordings of these webinars. So if you want to visit the social media platforms, you can do so. We also have a Facebook page. It's called Connect Conversations. So you can also visit those social media pages if you are interested in the webinars that we host. So we have a few future events that we are planning. One on the 9th of June, and then we have a course on manufacture and registration of biological and biosimilar medicines that will run from the 16th of June to the 21st of July. So if you are interested, please contact us on nano@pharmaconnect.co.za. So this is the course that I was referring to. This will be presented by Pharma Connect Africa in collaboration with the International Center for Genetic Engineering and Biotechnology. It will run from the 16th to the 7th of July. Like I said, it will be on the Zoom platform at around 9 o'clock to 11 o'clock South African time. So we will also be running some online courses. So if you are interested in them, those are the topics, and you can again contact us on the email address that is provided. So what we are here for today is to talk about radiopharmaceuticals. So I'll just quickly give a short background on the aim and purpose of this webinar. So we'll be talking about radiopharmaceuticals, and these are essential therapeutic and diagnostic agents that can be used for cancer diagnosis and therapy, and in some cases for inflammatory diseases. And so unfortunately in Africa, we have a burden of high cancer cases. And this is because most of the time have late diagnosis and poor access to diagnostics and treatment. And so if you want to use radiopharmaceuticals, like most of the radiopharmacists and radiopharmaceutical chemists know, you have to be able you have to be trained to firstly handle radiopharmaceuticals and how to use them safely as well. And so for today's purposes in this Connect conversation, we will also then explore how radiopharmaceuticals may be safely accessed and used on the African continent. So the first speaker up is Prof Jan Rijn from NEXA. He's a principal research scientist there, and he will be talking about clinical uses of radiopharmaceuticals. Thank you, Prof, for coming and agreeing to present to us. So you can share your slides.
Right, thank you, and thanks very much for the opportunity. I've perhaps deviated slightly from the title that you gave, Amanda, but I think it hopefully is nonetheless interesting to the audience. I'm going to take a little bit of a long-winded route. Sometimes it's called a scenic route, so perhaps you enjoy that. So when we're dealing with a pharmaceutical, there always has to be a reason why one does this, and that obviously relates to where the market is and what the market is dictating. So from the market intel study that's at the bottom here, you can see that what is expected from until 2030 is quite a substantial growth in the market. And we have to distinguish two things in radiopharmaceuticals: is diagnostic, that's the brown line at the bottom, and then the blue line is the therapeutics. And there was already a blockbuster that came to the market called Xofigo, bone metastasis treatment, and that certainly caused an inflection in the curve. Lutetium is just last year or in the process of being registered by Novartis, and then we expect more of these compounds to come online in a mass as new therapeutics, and that will then drive the amounts. And you can see the amounts here are in US dollars millions, so it's now becoming a big market. So also one needs to, and I'm more from the research side, so my presentation is more steered towards what is new and what's done in research. So important thing here is that once compared to conventional radiotherapeutics, how is a radiotherapeutic and a diagnostic doing? I think it's interesting stats is that there's a higher chance of success based on the statistics that diagnostics and even radiotherapeutics make it to the market other than conventional radiotherapeutic, other conventional therapeutics. And the cost is generally speaking also less. And the reason for that is that there's a visualization and imaging that's available. Someone is non-invasively all the time aware of what is your agent doing, so you're not going blind, as I will demonstrate. The development in our case through the clinical pipeline is slightly different. So on top, you see the normal, which everybody's used to. We have an extra step, of course, which is radio labeling. Preclinical, we are often allowed first in man before we go to full GMP manufacturers. Doesn't mean that we don't do this according to GMP practices and principles, but there is a phase 0 option which is allowed due to the very, very low levels of active compound that we're using. And that's what's unique about radiopharmaceuticals. We're dealing with the active ingredient, which is the radionuclide in the order of nanomole that is injected. And you can see some indicative numbers, how long it takes, and what the costs are. So what is a radiopharmaceutical then, just to elaborate on the basics? It consists of 2 compounds, the radionuclide and the biological compound. For my previous manager, the way that I had to explain this to him was to say, okay, so we're putting a GPS, which is the right-hand side, on the radionuclide, which then dictates where the radionuclide is going. So the radionuclide, of course, is the one that allows the external tracking through imaging instruments, or can deliver a radioactive payload directly to the area of concern. The biological compound is which has to be specific for organ. It has, of course, not to be, allows for the radio labeling, which is the combination with the radionuclide, and it must be as well non-toxic and ready for human administration. So nuclear medicine imaging, it consists of, and the difference here is depicted as a very old picture, but still very relevant. What's the difference between X-rays, meaning standard radiography, and nuclear medicine? The one is an X-ray is a static, stand in front of a camera. We get a picture of what is happening at a certain point in time, versus where you have nuclear medicine and radioactivity is injected, circulates, and it is then gives you almost a video, because you do this over time. You can see how this radioactive particle is moved around in the body. Often we have now, well, it's become standard now that CT and the imaging modalities are combined to give better images. So where does radioactivity come from? Well, there's generally speaking 3 sources. The one is the reactor, and that is the Safari 1 research reactor at Palandawa, 20 megawatt full tank type. And then there is a cyclotron at Itemba Labs. And also we can get this from generators. A generator is like a cow that you can milk at various points in time. It's generally done without any electricity. So this is a load shedding resistant piece of equipment. it uses evacuated vials to move the liquid through your column, and you then have your radioactivity milked out of this cow in inverted commas. We're quite unique in Africa. In fact, we are one of a handful of producers in the world, together with Nexan NTP. We are producing roughly 15 to 20% of the world market of the main leading isotope. And you can see where the others are situated in the world. Quite a success story. It's a 1 to 1.5 billion Forex income that is generated through these activities per annum. Sorry for more principles, but I think most people are aware of this, is that an alpha beam or an alpha particle is stopped by a piece of paper. Beta will travel through it, but be stopped by wood. Gamma, X-rays pass through wood, but not through lead, and the neutron passes through all of that. Now, if we translate that to what happens in humans and in vivo animals as well, you will see that a beta particle travels up to 100 cells. An alpha particle is between 1 and 10 cells that it will travel, and then Auger electrons travel just within the cell nucleus. For completeness, what is an Auger emission? It is a cascade of electrons, and in the interest of time, I'm not going to bore you with that, but it gives you very localized radiation. That's what we ultimately strive for. How do we design radiopharmaceuticals? Well, we're looking at a tumor, and we're looking at something that's very specific there. And often we look at the tumor-specific receptor sites that are overexpressed on a tumor and as not expressed on of course, healthy tissue. That's the distinguishing factor. Then we have a targeting molecule, and at the back end, we sneak in the radioisotope, almost like a Trojan horse. Sorry for more principles. What is PET? PET is positron emission tomography, and it is really where Einstein's E equals mc square has come to practice. So we have a positron, which is a positive electron ejected from the nucleus upon decay. It meets an electron, and you have analyzation of mass giving off 2 photons in opposite directions, which can be detected in a ring of detectors, which then provides you with an image. Now, these machines look very much the same like CTs, and they often combined with CTs, but the image is not looking static, but you're looking at specific hotspots. And in this particular case, is looking at use of glucose, uptake of glucose over standard or normal tissue, which indicates that there is could be a potential problem. Spect is the old gamma imaging that is still around, but has been made enormous strides as well in terms of the image quality. And here you can see what is possible these days. Here the spots will be higher uptake than normal as superimposed on a CT. So the CT provides you the background of the skeleton, and then the radioactivity is measured by the Spect camera and provides you where there is higher uptake. In this particular case, this is bone metastases. And then on the right-hand side, you have a lung perfusion study where the patient would inhale radioactivity, keeps his breath, and we take the picture, and we can see whether there's specific areas in the lung that will not is not reached by air. So this important part is this is all non-invasive. Moving into therapy, in the past we were using heart beaters, meaning heart in this particular case is highly energetic beaters that travel at least 100 cells far. It's almost like a shotgun approach. It's still used in thyroid cancer very successfully. It's 50, 60 years old, and it's still one of the products supplied every day by NTP to the country, to Africa, and to the world. There was also in the early 2000s 2 antibody-based radiopharmaceuticals called Zevalin and Pexar, but they've been declined. And I think part of the reason for the decline was the large range these particles were traveling. And because if you travel beyond the tumor, there's always healthy tissue. So we became smarter with that through research. And I think this is where this become the norm. And the theranostic approach is now very much cemented in nuclear medicine practice. So to illustrate what theragnostic means as a combination of therapy and diagnostics, is that one would have two patients here. Both are suffering from neuroendocrine tumors, and you would be using gallium-68, which is a diagnostic PET agent, coupled to a peptide that's specific for an overexpression of the somatostatin receptor on tumors that are coming from the neuroendocrine, which are pancreatic of origin in essence. And if you would do your imaging scan, you could see in this particular case on the left hand, there's no uptake. However, on the right, you could see this uptake, and therefore we can treat this patient. And by replacing, now the gallium is replaced by lutetium. And in doing so, you can see after one treatment, there is the number of lesions has decreased. So this brings, of course, to the fore personal medicine. We can do this not blind and not a one-size-fits-all. We know how the patient looks and we can use medical physics and dosimetry to calculate what the dose the patient should actually be getting. Just one more example, that these images are three-dimensional. So this is an actual almost cross-section through the abdomen, and you can see before a number of these lesions, and after treatment, except for the primary tumor, which is still there is very little metastasis left. And more of these images, so imaging before and after, these are both PET imagings. The middle ones are Spect images using the radiotherapeutic as the source of your imaging. So here you would see quite a substantive lesions in this particular prostate cancer patient. And as we then progress towards the right, this becomes less, and this patient in this particular case was disease-free. We've also been doing some work together with Steve Biko Academic Hospital, Prof Mike Soteche, on the use of other radionuclides. In this particular case, it was with bismuth 213, and you can see after one treatment for soft tissue metastasis also coming from prostate cancer, and here we will be using PSMA. PSMA is prostate-specific membrane antigen, so that's a very small peptide, but it's very specific for overexpression on prostate cancer cells. So this is the GPS. It takes the bismuth, which is an alpha emitter, to the site, and you can see how the PSA levels dropped and after one treatment cycle. Prof Soteche has certainly made headlines in the world of nuclear medicine, and so it's a proudly South African story, is that you can see here, he then switched to actinium, which is a quite a longer half-life isotope, also alpha emitter, and not only one, but several alpha emissions. And you can see here a patient that really had no hope at all of survival after a number of cycles. You can see how the PSA level dropped almost to normal, and the patient completely cleaned of his prostate cancer. So just to complete the story, what I've been saying, we're looking for pairs, because theragnostics means you need a pair with it. So lutetium is paired very well with gallium. There are upcoming scandium pairs. There is a lot of talk about copper 64 and copper 67, and particular research that we have done in the last few years between my group at Nexa, as well as the Paul Scherrer Institute in Switzerland, is on terbium-161. And I can just quickly show you how this looks in, and again, we're using prostate-specific membrane antigen as our GPS, our vehicle. And you can see how clean and clear the uptakes are in patients. In patients. This is preclinical work, of course, and you can see the uptake selectively in the tumor. Almost no other organ is affected, and I think that there's a lot of talk about that, and what's the space for improvement even further beyond what actinium is capable of doing, because terbium ultimately has a if I can just go back there to this slide, the difference here is terbium contains these Auger emissions, so very short range emissions on top of the betas. So it's really looking to also get rid of micrometastasis. Switching slightly gears is, and this is perhaps for a pharmaceutical audience of interest, is that we, in technecian, we've always used kits, and we would have used compounding of the kits in the radiopharmacy at hospitals or central radiopharmacies. But the question always asked, can we do that for PET? And the answer is yes. So this is a kit that was developed here, again, locally for the local generator produced by Timber Labs. And this is a shake and bake situation. You would elute the generator, put it in the vial, wait 10 minutes, and you're ready for injection. So often people ask, what is a kit? And it's none of the above. But it ties very nicely in with, which is important, I think, for the African continent, is that generators make things much easier, because you have a shipment once, and you can use, for instance, this gallium generator you can use over a period of 9 months, and even therapeutic generators are now available, and this one will last roughly 10 days, and you can use this on frequent basis. That would circumvent the logistics of supplying radiopharmaceuticals on a day-to-day basis, which is, of course, the norm in the first world, where the logistics are a lot better. But I think in the African context, the generators and the kit combination is something that we should be looking at. I think I don't want to go on too long. I'm going to then try to summarize on this, and then I'll just mention a few things, because I've only talked about oncology, and one should know that there is beyond oncology also use in infection imaging. So just to summarize, what is important when we're dealing with radioisotopes, we should have them no carrier added, and I'm not going to dwell into that too much. We're moving towards shorter range emitters. That generators, I think, the way to go, and that comes together with the demand for kit formulations, and we would be compounding at point of care. And theranostic pairs are the ones that we really should be looking at in the future. We also, just to complete the story, is that there's also an option to use malaria antibodies to label what's going on in specific patients that have problems with malaria, and one can obviously use non-invasive imaging as a tool in drug discovery for it. So what we should illustrate here is that we have an existing toolbox with radio tracers in, and these can be used as drug discovery. And we should always aim to direct the radio label and analog of a hit compound. And for us, because F18 is such a good radioisotopes, we would be interested always in F18 containing compounds, because we can replace the cold F with a hot F, and then you can do a lot of things. So I am going to just stop and say this is, of course, not done by that one person, but there's many teams around, and this is not the last pictures. There are many, many more. And I would like to then stop with the famous Marie Curie, with some quotes from her. I'm handing back to you, Chair, and I'm willing to take any questions if there is time. Thanks.